Evening, everybody, and a very warm welcome to you for the third of our series of webinars for biology teachers, and the first one of 2026. My name is Emma O'Donovan. I am a head of post-primary sales here at Folens. And this evening we're joined by authors David O'Brien, Jennifer Groves, Stephen Murphy. And looking back over the last few webinars that we've had for biology, they've been a massive success. We're leading on from what we've done previously in where we introduced the webinars by doing a 5th-year plan. We introduced David's web page, and we are also David went through more difficult subjects like respiration and photosynthesis. So delighted you can join us this evening. It's a difficult thing to do after a full day's work. Just a few housekeeping things first. If you are joining us, to turn off your notifications, if you click your profile picture in the top right-hand corner and set the status to do not disturb, and then all the team notifications that come in will be silent, so you'll be able to add then to the chat, pops up to the chat if you need to, but there'll be no notification pinging throughout, so it won't be a distraction. So this is a webinar to launch our textbook, Biology in Practice, and worth the wait is the tagline. And worth the wait is for us as a publisher, we waited for the SEC sample paper so we could get it streamlined to the new specifications. So we hope that you will find this a very, very useful wait in that we'll have a textbook that won't have too much in it, but we'll also have a textbook that will have more than enough for this new specification. It's very much aligned to what the exam is looking for. We've had the luxury, I suppose, of having the SEC sample paper before we wrote the book. So there's a lot of material in there that is very, very streamlined. The package already, we have the Biology in Practice exam prep book, which is available previously. It was launched in November, and it is literally a bank of over 200 pages of exam prep questions. So these were based again on the SEC sample paper with a special sign for Nature of Science questions. Just in the warehouse since last week is our investigations book. David will be going through that and he will show how it's aligned to the investigations. And then the Biology in Practice textbook, I'm delighted to say, went into print last week. So we should have it in the next 2 weeks or so, and the reps will be going around with copies of that. So I'll have a QR code at the end of this where you can contact your rep if you're looking for copies of that or you'd like to be considered to get copies of any of those. If you're looking for the exam prep book or the investigations book, our reps are currently going around to the schools with those now, so they'll be ready available. So just have a quick run through our profile of authors, for those of you that haven't attended these webinars before. David O'Brien is Assistant Principal in Ballincollig Community School. He's teacher of biology, science, maths. He's an ISTA biology convener, and he's the co-author of the Essential Science programme, and he's an SEC examiner. Jennifer Groves is Head of Science and Biology in Blackrock College, and she's experienced in international curricula and as also an SEC examiner. And then finally, Stephen Murphy is the Science Education Lecturer in UCC, and he was a former biology and computer science teacher in Gaelcholáiste AG. So you can see it's a textbook that's written by practicing teachers for the classroom. So we realise straight away that this textbook will be right on target. Just to make you aware, for those of you again who didn't attend previous webinars, our seniorcyclebiology.com has got a huge following in that it's divided into 3 areas. It's fabricated by David O'Brien, one of our authors, and the new biology specification for 5th year, he goes through all of that. He gives tips and tricks for it. He also has some tips and tricks for the old biology specification, some past exam papers, and for junior cycle science. He's also available on Instagram at Senior Cycle Biology for any tips and tricks again that he's finding in the classroom that he's like to share. That is up there. So if you want to follow him on Instagram, please do. Just to give you an overview of what we'll be covering this evening, David will lead the webinar with the Biology in Practice investigations. So he'll be going through the book that we've launched, but also how this ties in with the investigation. We'll do an overview of the textbook. Stephen Murphy will lead that and go through the features of the chapter of the textbook, just to show you how aligned it is to the SEC sample paper and the NCCA curriculum. Questions at the end of the chapter, then Jennifer Groves is going to go through a few of the questions, and she'll also share a summer test with the marking scheme for that, which we'll release at a later date. And then David is going to go through some SEC exam questions as well. So it's a fairly packed evening. We'll have a Q&A at the end, and hopefully we'll get through it all in the space of 40 minutes. We do have a little raffle, and we have a survey if you wouldn't mind filling that out, but there's also a little prize for that, so it might be a nice way to end the evening. So the giveaway for the attendees is a class set of biology and practice and exam prep book, the raffle for that. And then you have the survey, which is a one for all, a €100 one for all voucher. So might be a nice end to the evening for some of you. So with that, I'll pass you over now to David O'Brien. So David's going to share his screen now, and he'll take you through the investigation. Thank you very much.
Hello, everybody, and thank you very much for attending tonight. Emma, you might just confirm, please, that you can now hear me and see my screen.
Perfect, David. Yeah.
Thank you, Emma. So, hello, everybody. What I'm going to take you through is I'm going to take you through the investigations book. Now, both the investigations book and the textbook for biology in practice are available to view on the Folens website as an online file. So I would encourage you to do this after the webinar. Our investigations book is divided into 3 sections: Section A, Section B, and Section C. Section A is the first part of the investigations book, which consists of all 26 of the investigations specified by the National Council for Curriculum Assessment in the biology specification. And we have areas in which students can practice writing laboratory reports, analysing data, answering questions on those investigations, and of course QR codes and video links to each of those mandatory or specified investigations. Section B in the investigations book is all about honing and creating and developing our students' skills. It's an entire section simply based on secondary data, where students are given data in unfamiliar contexts based on the core strands in the biology specification, the structures and processes of life, the interactions of life, etc. And students then evaluate, interpret, synthesize, and reason the data that they're given. Section C is based on the biology in practice investigation, this laboratory-based research project, and I'm going to take you through each of the sections now. So, Section A, we've 26 mandatory investigations, and there's a full set of instructions and templates for each. QR codes to video to each of them, and this will really benefit those who will be doing primary data for some variables and secondary data for others. Highlighted in every single investigation is the dependent variable, the independent variable, the control variables, how you ensure fairness, accuracy, precision, and reliability. Quantities are given for each investigation, which is important because, of course, students may be designing their own investigations, and really important is that in every single investigation, we've incorporated a reflective approach. So we have a detailed procedure with diagrams. We have this introductory planning, identifying the variables, selecting suitable equipment, drawing diagrams, recording their method, and now we have the data collection, data analysis and conclusion. And on the right hand side of the screen, you can see that every investigation includes a reflective approach, and this will stimulate students and give them plenty of practice at looking back on their experiment and what would they do differently. Here's another example about transpiration of plants. Again, it is the same layout. The beginning is planning and preparation. We then have recording of data, analysing of data, and then, of course, we have this reflective approach, which over time, the more practice the students have of it, it will develop their reflective skills. Now, the specification requires, as well as that, secondary data to be gathered from scanning electron microscope imagery. And you can see that we will support you in guiding the students to achieve that learning outcome, because we have secondary data of scanning electron microscope imagery, and they have to identify the various organelles. On the right-hand side of the screen, you can see when students are asked to do calculations, for example, calculating abundance of species, calculating distribution of species, calculating species diversity values. These are all related to ecology. Loads of space is given for the students to practice their calculations and to fill in their formulas. In addition, in Section A, after all of the 26 experiments, students are then given questions based on the experiment, and this will further ensure their understanding and deepen their understanding of these 26 investigations. Whether it's primary data or secondary data, students can still develop their knowledge with these questions. So that's Section A of the investigation book package. And now we're going to look at Section B. Now, Section B is 26 pages dedicated to secondary data, tables, graphs with accompanying questions, and this section is so important because it will benefit our students in two ways. This skills-based section will not only benefit our students in the final exam, but also in any research-based tasks that they may have to do. So this is just a snippet. These are 3 pages. Students are given secondary data. For example, there's one there about transport across membranes with different sucrose concentrations, and they have to calculate the averages and describe the relationships and estimate the approximate percentage change in mass. In the center of the screen, you can see one that's to do with the growth rate of microorganisms over time, and they have to match each description from row A, B, C, or D with what is happening in V, W, X, Y, and Z on the curve. And on the right-hand side, then, we have a graph related to recombinant DNA, which is, of course, genetic engineering. And again, students may not have seen or carried out any investigation with the optical density of cultures, but by giving the students the data in unfamiliar context, we can really prepare our students for any challenge that can appear, not only in the Leaving Cert examination paper, but any research task which they may be expected to do. And of course, this ties in essentially with the overall aims and objectives of the unifying nature of science strand, where we get students to evaluate, to critically analyse, to identify anomalies, to understand if a source lacks bias or not, and to draw and justify conclusions. All of these learning outcomes can be achieved in Section B of the secondary data section. Now, here's again with the osmosis one that I showed you. Again, more with osmosis there. There's a plethora of data analysis questions, 26 pages simply on secondary data. Section C of the investigation book, well, of course, we based this section on 2 parts. First of all, the guidelines that were produced by the National Council for Curriculum and Assessment, and as well as that, the sample and live documentation which was produced by the State Examinations Commission. And we have broken down the steps into 6, the steps into student-friendly manners. We've given advice on how to present data. We've given them advice and practice on researching, also on referencing, and the checklist. So the long NCCA document has been presented in a student-friendly manner in which students could follow. Of course, this really reduces the cognitive load on students and allows them to make sense of the complicated process. As well as that, in this section, not only do we give advice, but of course, advice is one thing. We give them advice on how to write research questions and how to write hypotheses. But in Section C, we give them practice. So students are given research questions and they have to formulate a hypothesis based on those research questions. So they have the advice, but they also have the practice. Students, of course, when they're carrying out research projects, will be expected to present data in various forms. And this guidance in Section C can help students to decide which graph best or which data presentation method best is used to present the particular data that they have collected. As well as that, referencing is a big part of the NCCA guidelines. Not only on the left-hand side have we followed the guidance of the NCCA, but if you look at the right-hand side, we have provided space for a low-stakes practice at referencing. So before one might begin referencing, they're able to practice referencing in a comfortable, low stakes environment in their workbook, in their investigations book, where they're given particular sources and they're asked to write the reference entry and the in-text citation. Again, you've different from the World Health Organization one there, and then the last one, you've marine biology and ecosystem approach. We're giving them practice at referencing. Now, we all know, of course, that when the sample and the live brief came out, we were told about the different criteria in Section A, Section B, and Section C. And what I've gone through with you, okay, the hypothesis and research question, evaluation of secondary data, referencing, that's all Section A of this criteria you can see on the left-hand side. As well as that, if you look at those words, experimental design, safety, fairness, selection of equipment, accuracy, we've covered that in all of our investigations in Section A. And as well as that, if you look at D, okay, down there, you've this reflective approach which must be incorporated. Now, in a student-friendly manner, we have deciphered the NCCA guidelines and this SEC documentation to produce a student-friendly checklist for any research investigations that any student could do. And again, this checklist is in the investigations book, and it can be used by students to manage their workload, to support their work, and to really help us as teachers as well identify what is being done well by the students and maybe what have they yet to do. So I've just taken you through the format of our 3 investigations book, or our 3 sections in our investigations book, and I'll happily pass you over to Stephen, who's going to talk now about the textbook. Thanks.
Hey everyone, just give me 1 second there now. Sorry, no, technical problems. Oh, there we go. Can I just confirm, Emma, that we can see this and hear me?
Yeah, good to go.
Super. Hi everyone, my name's Stephen Murphy and I'll be taking you through the textbook of Biology in Practice by Folens. So the textbook was designed in both a thematic and pedagogically sound order. So like, for example, we would see that enzymes are to be taught before respiration, and nucleic acids would be taught before viruses. And, you know, it makes sense because respiration is so heavily influenced by enzymes, and that viruses, you know, you'd be talking about DNA and RNA viruses, and you need to make sure that the students understand what DNA and RNA is and the differences between them before you start talking about a virus that contains those types of nucleic acids. In terms of the structure of each chapter then, the learning intentions are linked to the learning outcomes. And, you know, I find that the learning outcomes here are great for visual learners because they have that traffic light self-assessment section at the end where students can fill in where they are on each of the learning outcomes, which is very good. And, you know, it's nice for students to reflect upon their learning as well. At the start of each chapter, then, we talk about the connections that that particular chapter, for example, cell division there, has with other chapters. And that's reflected with the that's reflected in the SEC papers as well, where the questions are quite thematic and they draw from a lot of different chapters in the different parts of each question. And then these chapter connections and these learning outcomes that are presented at the start of the chapter, then they are reinforced at the end of the chapter with connection questions that asks students to draw on their knowledge of other chapters to answer questions about that particular chapter. So we can see there about the cell and cell division, you know, suggests why ribosome numbers increase during interphase. So again, it's the students drawing on their knowledge of ribosomes and cell structure when answering questions about cell division. And we feel that that is reflective of the SEC sample papers. And here we see about plant structure in cell division as well, just some quick secondary data analysis, a nice table there that students need to interpret, and a link between that cell division and genetics. So some of them are kind of obvious, like maybe cell structure and genetics with cell division, but then some of them are not so obvious, like plant structure. And, it gets students seeing biology as a whole rather than, each individual chunk of information contained in the chapter. Throughout each chapter, then there's a kind of a concise summary provided at the end of every chapter, and those the higher level content is clearly indicated, and this summary then is highly connected to the definition boxes that are found in each chapter. So, students who are kind of coming towards an exam or an assessment, they can quickly find the key definitions that are required by in an assessment. We find as well that it was really important that our diagrams are both detailed but clear, and we feel that the diagrams are reflective of the diagrams found in the State Examinations Commission sample papers. So we feel we found the right balance there between complexity and clarity. I'd like now to discuss the nature of science and the cross-cutting themes in terms of our textbook. So it's really important that nature of science is not taught in isolation, that it's not taught at the start of 5th year and then parked away while other topics are taught. Nature of science permeates and is integrated into every topic in biology, the new biology specification. And we reflect that both in the nature of science boxes that we embed into every chapter, and also our experiments. Students can identify how scientists work, how scientific ideas are modified over time, and they also gain an appreciation for models. But things aren't forced. The nature of science is integrated very naturally where appropriate into the specific topics and chapters. This is important because 30% of the SEC exam paper questions are based on the nature of science, and we clearly highlight in our textbook where the nature of science questions are, that students can practice these types of questions. In terms of the exam preparation book, then, as I said, all the nature of science questions are clearly marked in both the textbook and in the examination preparation book. So all students need to look for is that little NOS green icon, and they will find nature of science questions to practice with. We also integrate the 3 cross-cutting themes into the textbook. Now, it is done to reflect the State Examination Commission's sample papers, that these nature of science boxes, these cross-cutting themes boxes are sprinkled across the chapter, and each chapter has one of these. It was our hope that by integrating them gently into each chapter, that that students won't be thrown by them on the day of the Leaving Cert exam, or if you present them with a question like this in the summer exam or Christmas exam. Jennifer, I think that
Yep.
You're up.
Hi, everyone. Good evening. So I'm just going to talk about the questions that we have at the end of our books. So, Stephen, if you could put your screen
Yep.
on big, great.
Mhm.
Thank you. So, first off, we have exam tips throughout the chapters. And if any of the chapters have experiments as well, we've put in some exam tips that we've put in there for the experiments as well. So we have exam tips throughout the chapters that are there. When we're putting the questions together, we've done the questions, like 4 main types of questions that you'll see at the end of each chapter. So the first one will be the questions from the State Examinations Commissions. And what we've given is some model answers throughout, and you can see that they're there in blue. They're highlighted for the students and for the teachers. The next type of questions that we have are revision questions. So the revision questions at the end of each chapter are based on the textbook material, and they are to cover the core knowledge for the students, so that before they go on to exam style questions, that the students have the material understood. If also there's any investigations within the chapter, as you can see here, we have enzymes. We've also included some revision questions based specifically on the structure, the procedure, the variables for the investigations. And as mentioned before by Stephen and by David, we've also integrated there the nature of science. So these questions have, we've identified structure. So using keywords here as well, where the students have to identify, explain, describe, interpret. So what we're trying to build upon is we're trying to build upon students' progression here. The next questions that we have that come up are exam style questions. So when we all saw the sample papers by the SEC, we could see there was lots of patterns, but basically we were where the students have to interpret data, evaluate, look at reliability, recognize variables. There might be some articles or extended responses. And again, all of this is linked in with nature of science. So we wanted to make our exam style questions incorporate all of that. So again, we progressed from our revision questions and we're leading on to our exam style questions. And the last questions that we'd have at the end of the chapter would be our connection questions. So we don't want to compartmentalize students with just topics that they have to know, for example, enzymes or respiration. So this new specification encourages the integration within the strands, the integration of the chapters. So we want to prepare students for that. So no matter what chapter the students you've covered with your students, there will be connection chapters linked in with that. So what we're doing is we're preparing the students so when they do see their paper in 2027 that they're not going to be surprised, and hopefully there'll be no stress there, for the students. And then if you want more questions, just as David and Stephen have said, we have more questions and examples of style questions in our exam prep book. And again, we're building on our students' exam technique across 10 formats here. So the formats that we're looking at, Stephen, if you click the next button, thank you. So all of this is going to be assessed. So we're assuring a balance through the students' preparation. So we're looking at their knowledge. They'll be assessed on the nature of science. They'll be able to label diagrams, draw diagrams, complete tables, interpret tables, analyze graphs, draw graphs, and write extended detailed answers. So we've got quite a lot of questions available for you guys to use. So what we've also done is we have made our scheme of work for the second term for fifth year available to you guys. So the chapters on our scheme of work, Stephen, if you click on, please, thank you very much. So the chapters that we have there are fungi, bacteria, plant structure, plant transport, nucleic acids. I'm trying to remember. So what we've done is we've made hopefully what you think as well is a professional approach to the summer exam. So the layout will be similar to what we shared with you guys at Christmas. There's 6 questions going to be in total. Question 1, we said, is compulsory. And it's going to start off with a short comprehension. So it's mirroring the SEC sample. What students will see or what you'll see through here is we have secondary data available. We have the nature of science embedded throughout. Students have to draw diagrams, label diagrams, interpret data, draw graphs, and extended answers. So this will be available to you later. It will be in a Word document. So if you want to use some, you can use it. It's totally up to you. We're just hoping we ease some pressure for your summer exams. There will be a marking scheme available also, and the marking scheme, what we're doing, we've made the marking scheme and we've set it in line with previous marking schemes by the SEC. So that will be available for you guys to use also. So now I'm going to pass you over to David, who's going to talk a little bit more about sample papers.
Hello, everybody. I'm back. So we're going to look at how fortunate we are in Biology in Practice that we waited for the SEC sample papers. Because we waited for the State Examinations Commission sample papers to higher level, to ordinary level, this has informed our writing, and we've been able to tailor our writing to match the precedent set by these examination papers. So just to show you, we are all very familiar with, from sample paper 1, the model of the fluid mosaic model of the cell membrane. And of course, students have to label the parts of the cell membrane. And as such, as you can see, how do they master this? Well, we have used the exact same imagery as the sample examination paper. Students are able to identify molecule A, identify molecule B, and identify molecule C. We've got you covered with regards to that. As well as that, Stephen mentioned earlier that the nature of science is not taught in one big chunk in isolation at the beginning, because that will cause a lot of time to be taken up. In every single chapter, as you and your students come across various phenomena in the nature of science, you will be introduced to them. For example, we bring in qualitative data and quantitative data in the food tests, because that will be the first time they encounter it. Again, we bring in the development of scientific ideas and technology in science in the chapter on the cell, because of course, that's where we encounter that concept. Another sample paper that came up was based on identifying the different types of biomolecules based on their elemental composition. And of course, this informed our writing. And you can see that, you know, proteins could be CnH12N4O3. Again, they're used to identifying lipids because there's no elemental fixed ratio, but they do have carbon, hydrogen, and oxygen. And as well for carbohydrates, they have that ratio. So the knowledge of the sample papers have informed their writing. And as such, any student who uses this textbook will be able to answer every single sample paper question. Let's take a look at photosynthesis. And of course, in photosynthesis, we were given this diagram which represents the reactions that happen in the thalloid membrane during the light-independent stage. Now, as such, it was very important to us as authors that our diagrams were straightforward, our text was clear, call-outs were used, because as you know, the students in front of us, okay, their reading ability, as we have seen over time, has changed. And so to reduce the cognitive load, okay, our book really reflects this. You can see, of course, part 1, what's the function of chlorophyll? And we have the chlorophyll absorbs sunlight energy or light energy, and this energy is used to energize electrons. And you can see on the right-hand side, a snapshot from page 125 of the book, we have the light energy on the right-hand side traveling from pigment to pigment. Part 2, it asks about photolysis. Now, I can't stress how important this is, because in the specification on page 54, it talks about photolysis due to a charge difference, okay? The specification, it's a higher level only learning outcome, and it mentions photolysis occurring due to the positively charged chlorophyll molecule. And of course, we've got you and your students covered. Photolysis, of course, the breakdown of the splitting of water into protons, electrons, and oxygen, and the electrons are then attracted to the positively charged chlorophyll to replenish the stocks of chlorophyll. But it's this charge difference that drags the electrons to that chlorophyll molecule within the membrane. Part 3, the final electron acceptor, again, NADP, we have the definition highlighted. It's broad, it's a call out, it grabs the student's attention. Part 4 described the role of molecule B, and of course we all know molecule B is NADP, and of course what's its role? Again, if you look visually, okay, we've tried to explain that NADPH and ATP comes from the light-dependent stage, and NADPH, of course, provides those hydrogen ions, those protons and electrons to join with carbon dioxide using the energy from the splitting of ATP to form glucose. Part 5, they're talking about that enzyme, and of course that enzyme is very important. It happens in both the light-dependent stage and the light-independent stage of photosynthesis, as well as stage 2 of respiration. It's ATP synthase. So any question that comes up in the sample paper can be answered with us. Again, we have a thematic question, which was based on classification and as well as that genetic engineering and evolution. Now, Thermus aquaticus, this bacteria, which contains the heat-stable version of a DNA polymerase, of course, this is what came up in the sample paper. And again, on the right-hand side, we have the biology in practice, and you can see we've explained how to analyze these phylogenetic trees, what these branching points mean. Similarly, in human reproduction, we were given a graph about tree hormones. We've went even further, okay, and we've included more hormones, okay? We've included an additional hormones so students can analyze what happens during implantation and birth and the menopause and so on. You know, the red arrow on the left-hand side, that'd be progesterone, and B would be oxytocin, C being prolactin, and of course, on the right-hand side, we've the biology in practice one. Not only do we have the hormones identified, but we have their role and what influence the hormones have on female reproductive system. What we were really lucky, the fact that we waited for the sample papers, was really about chapter 15. And chapter 15 in our book, it's called the Manipulating Genomes. Now, I'd just like to take you through what came up about DNA profiling. Now, of course, they were asked about, you know, what's happening during the denaturation stage of DNA profiling, specifically polymerase chain reaction. PCR is made up of 3 steps: denaturation, annealing. Annealing is also called cooling, and then extension. Now, as such, if you look underneath the box in the middle of the page, it says some components of PCR include a DNA template, dNTPs, primers, Taq polymerase, Taq polymerase, that's the heat-stable DNA polymerase found in Thermus aquaticus, pH buffer, magnesium chloride, and thermocycler machine. Now, all of that language must be understood by students. And in our Manipulating Genomes chapter on PCR, DNA profiling, and genetic engineering, we've covered that. We've our double-stranded DNA, we've our heat-stable DNA polymerase, we've our dNTPs, we've our primers, or pH buffers, and as well as that, we have our magnesium chloride. In the sample papers, we were asked to describe what's happening here during gel electrophoresis. And again, this shows that we have taught students in our book how to analyze DNA profiles, which of course they are expected to do from secondary data. So the full package, I've just taken you through the textbook, includes more than just the textbook, the exam prep book, and the investigations book. Every single chapter has interactive PowerPoints, and I'm just going to show you one of the PowerPoints now. Very briefly, this is one on enzymes. Now, our aim when we were writing the PowerPoints was to, again, I'm using the word reducing the cognitive load, because students in the sample paper are given stimuli. They're given pictures, they're given graphs, they're given tables. So we reflected that in the PowerPoint, okay? Definitions are highlighted, images are large, text is kept to a minimal. It's the concept that we're trying to get across in the PowerPoint. There's no need for loads and loads of text. You can see the induced fit model, and you can see at the top that's noted as higher level. So all of that work is done for you. And inside now our PowerPoints, we've included links. I'm not going to click on it, but you can see it's a hyperlink, which will bring you to short YouTube videos, which you can use while teaching your lesson. Graphs are labeled clearly in the PowerPoints with callouts to explain exactly what is happening to the independent and dependent variable at that particular point in the graph. Similarly, again, text is kept to the minimal, and as you can see, I've just very quickly show you these in the PowerPoint. Every single experiment has a scan me. So if the students scan the PowerPoint, or you scan the QR code, or you scan the QR code, it will bring you straight to a video of the author team carrying out that particular experiment, which will be useful for revision. Again, we've all these an analysis of graphs and finishing off summary. So in the full package, we've interactive PowerPoints, QR codes. Now, number 3 is BioBoost videos. Now, as we all know, in this new specification, there are some topics which are not only new to us as teachers, but will be especially challenging to students. So what we have done in our book is, for various topics, I have created what we are calling BioBoost videos. This will require students to scan a QR code, and it will bring them to a link which contains videos of me explaining difficult concepts such as respiration, photosynthesis, genetics, DNA profiling, epigenetics, etc. So I'm just going to play one for about 15 seconds so you can see how it works. And this will really help your students with revision. So when they open up the respiration chapter, they can scan the QR code and there'll be various videos on respiration.
Just to explain, there actually is audio with this, but I would imagine that on the Teams there's a little bit of a blocker with it, so audio is
no problem. Thank you for telling me that. Now
I'm
back
so
now.
There is actually an audio
So
script.
will we play it
You
again?
can hear David's.
Will we play it again there, Emma? Would you just confirm
just
to
a
me?
few seconds, yeah.
And as well as that,
Perfect.
within the inner membrane of the mitochondria, they have electron acceptors. Together, these protein complexes and electron acceptors are referred to as the ETC, the electron transport chain. Above the inner membrane is the intermembrane space, and then above that as well, you have the outer membrane surrounding the entire mitochondria. So NADH has been made as a result of the citric acid cycle.
So as you can see, these videos are on the difficult topics for students and teachers to help develop the conceptual understanding of them. Our teacher's guide, another part of the package, has all editable planning resources, which you can download and keep as they are or tailor towards your school. Scheme of work, template risk assessments, advice on extensions for experiments, and extra information for you, the teacher, to support you in teaching the topics. The questions in the textbook, the exam prep book, and the investigations book, the fully worked solutions will be available as well with the teacher's guide online on the Folens website. So I'm going to stop sharing now, and I'm going to pass back over to Emma.
Perfect. I'll just share my screen again there now. Sorry, give me 2 seconds. That was very well done, David, in a short space of time. It's pressure when you've got a certain amount of slides to get through, but well done. All the authors actually did a great job. That's just to explain the BioBoost videos. I don't know if you can see my screen there now. Yeah.
We can see your screen. Oh yes, that's brilliant. You can show the attendees that. Well done.
Okay. So what are you seeing on my screen now, actually?
We're seeing the thylakoid membrane.
Oh, right. Okay.
So
share the screen there now.
Yeah.
All right, give me 2 seconds there now.
There's no problem. It's great to have a live audience anyway.
So you can see the BioBoost videos there?
That's it. Yeah, that's the QR
Perfect.
code that will come up on the textbook. Thank you.
So
Emma.
that's just to mention that the BioBoost videos are also available in the PowerPoint. So when the teacher is actually teaching inside in the classroom, there is a QR code to the BioBoost videos as well. So the teacher can play that little video inside in the classroom. And when the student goes home and is coming across that difficult topic like the thylakoid membrane, that they can actually access those BioBoost videos as well themselves and repeat what they have learned inside in the class. So they're a really useful addition. They're new. We're hoping that they will be a huge success. I would imagine they would be, because the reaction from going around to the schools and letting people know of their availability is phenomenal. So we definitely think the whole package is worth waiting for, in terms of we realise that you as teachers had to wait a year or a couple of months at least, but we've waited hugely because of the SEC sample paper and aligning the whole programme to the to the SEC paper and the requirements for this new specification, we definitely feel it's worth waiting for. So we've had a few questions in during the week when people registered. So I'm just going to go through a few here now. One was when is the date for receiving a hard copy of the book? The actual hard copy of the book will be available soon. As I said, the investigations book came into the warehouse last week. So the exam prep book, if you are looking for copies of it, you can contact your local rep, or the investigations book, which the reps are going around. But the hard copy of the book should be available in the next 2 weeks or so. So as soon as they're in, the reps will be flying around to the schools with that. And we'll hope that, as I said, it'll be worth the wait. There's another question here. The spec mentions that a knowledge of the central nervous system is needed. And what exactly do students need to know? David, do you want to take that one?
Okay, yeah. So the specification mentions in the central nervous system, on the left-hand side, it says, you know, when the student learn about column, it says central nervous system, brain and spinal cord. Now, that's what it says. So I would definitely teach the structure, the parts of the brain, and I would also teach the spinal cord, you know, the ventral root, the dorsal root, etc. That's if it's in the student learn about column, brain and spinal cord. Thanks, Emma.
Perfect. Regarding the BioBoost videos, I just have a question here I'll answer. And there's one per chapter or one video for multiple videos. There is, at the moment, we have 1 or 2 per chapter, but that'll vary on the topic, and it'll vary from chapter to chapter. So it's not specifically tied to 1 per chapter. Another question here. Will there be a suggested marking scheme provided with the test? Jennifer, would you like to take that?
Yeah, there will be, and again, it's in line with the previous SECs. I forgot to say, guys, that each question we have followed the SEC samples where we've made each question 50 marks, but we have not allocated where marks should go. So we're leaving that up to you. But yes, there will be a marking scheme available in line with the paper.
Perfect. Another question. What about project checklist? Is there anything that could help here? Stephen, do you want to take that?
Sorry, with what do you mean by in terms of help? Like,
is there a checklist, or is there a checklist?
There is, yeah. So at the end of the exam, or at the end of the lab book, there's a checklist that students can use to check their own work at the end of each section, and they can make sure that they're on track for, you know, as the teacher sets mini checkpoints during the project, that they can, you know, see how they're getting on and making sure that they're meeting those soft deadlines set by the teacher.
Oh yeah. So I think, just to answer that question there, the person who asked that question is obviously asking about section C, Emma. Am I right?
Yes, I would imagine so, yeah.
Okay. So if you're asking about a checklist, okay, the best way in which you can get your students used to the nature of science is, at the end of every single investigation, is to do the reflective approach. If you start doing the reflective approach from the very beginning with the food tests and the microscope and the enzymes, which it's hard to believe we'll have another 50-year group in September, that's how we can harness these skills. Like, I understand what you're saying when you say help, but like, I suppose that's what I would encourage, this constant reflective development of skills, which are in section A and section C of the investigations book. Now, I just see another question come in there. Is there access to textbook and investigations book online? Yes, both the textbook and the investigations book are available to view online on the Folens website.
Yeah, and India has just put up the link there for Folens, www.folens.ie, where you can see a digital copy of the textbook, the investigations book, and the so that's in the chat there at the moment. There's another question here. Should you cover the epigenetics under DNA, David? Does that make sense to you?
Yeah. So if you're covering epigenetics, I suppose with DNA, Emma, is it?
Yeah, okay. That's the question.
So,
should you cover epigenetics under DNA? Yeah.
I would not study epigenetics when you're studying like the structure of DNA, because of course epigenetics refers to, you know, the turning off or the turning on of genes, but I would cover genetics, Mendelian genetics, whereby you study the inheritance of genes from parents to offspring first, because how can you study the switching on or switching off before you would study inheritance at all? So I would definitely do it after you've done crosses, for want of a better word, after you do crosses.
Perfect. And there's another question came in there about the webinars, previous webinars conducted by us, which would have been in November, or August and November. They're available as well on www.folens.ie. So those recordings for the other webinars. This particular webinar is being recorded and it will be sent out to you in the next couple of days. I think we're nearing the end of it. If you're interested in getting in touch with your rep here, I've just popped up the QR code. If you want to get in contact, just put in your roll number or your name of your school and your actual rep will pop up. And I think we're bang on target, maybe a little bit over. I pulled somebody from the raffle. So the raffle was for a class set of biology and practice for your school, and from St. Angela's School in Waterford, Sive O'Connell Lacey, and we'll be in contact with you for that. And the last thing then is, just to end the survey, a quick favour is we have a short survey that we're going to send out to you. Sorry, just to end the webinar. A short survey we're sending out to you. We'd be very much obliged if you took a minute or two. It's only 7 or 8 questions to fill it in, and hopefully it will result in one of you winning €100. So that person will be drawn in the next day or two, and we'll get in contact with that. So, on behalf of everybody here in the Folens team, on behalf of myself, India and Katrina in the marketing department, in particular India, who's been working all day on this webinar, and on behalf of the authors, a very, very, very big thank you to all of you who attended today. It's a big ask asking you to attend something like this after a full day at work, where you have loads going on in the classroom. So we're really grateful. We will have copies of the book in the next, I'd say, 2 weeks. So as soon as that's available, we will be out to you. And on behalf of me and the team at Folens, I'd like to thank everybody for attending today. Like I said, any of the previous webinars we had in Folens are available on folens.ie. And Stephen is actually giving another webinar on photosynthesis in the Cork Education Support Centre on, I think it's Tuesday, is it, Stephen?
It's David.
Yeah, oh, sorry, David. David is giving it on the Cork Education Support Centre on Tuesday the 10th, is it, David?
Yeah, this day week on photosynthesis. If you'd like to come along, I'd be delighted to see you.
Perfect. So again, on behalf of Folens, thank you very much again for attending. As I said, it is a big ask on a Tuesday evening, and we'll be in touch shortly with the winner for the survey. So thank you very much, guys.
Thank you. Bye-bye.
Thank you.
Bye,
Bye-bye.
everyone.